we'll be studying applications of paper chromatography now. So girls, you can separate many biomolecules like carbohydrates, then you have amino acids, then you have different plant pigments, then again you can separate different types of chlorophylls. In chlorophylls also you can separate different types of chlorophylls. Then you can check the purity of the sample. You can check the purity of sample. You can monitor the progress of reaction. So girls, now let us see the first application in detail. Simple. Suppose if you have mixture of carbohydrates, if you take simple sugars like sucrose, glucose, galactose, lactose, fructose. If you have all this, sugars in a mixture, you can simply do the paper chromatography on it, and you can use butanol, acetic acid and water in the ratio of 4:1:5, and you can separate this set of mixtures. Girls, these sugars are colorful or they are colorless? They are colorless. Then how to detect them? You spray the detecting agents. You can spray molish reagent, girls. For the sugars, you can spray simple molish reagent or Benedict's reagent, and you can locate this spots of this carbohydrates. Then, if you take amino acids, for example, like tryptophan, lysine, glycine. If you take this mixture of amino acids and you can do the paper chromatography on it, and you can detect them by spraying anhydrine reagent on it. For example, if you want to separate chlorophylls, chlorophyll A, chlorophyll B, etc., you can do that, and you can directly observe the spots because they are colored. And also plant pigments like xanthophylls, carotenoids, anthocyanins, they can also be detected. Now, what is the purity of sample? How can you check the purity of sample? How are we going to check the purity of sample? How can we check the purity? What is purity? Now coming to purity. So in the pharmaceutical industry, they will be producing many drugs. So you can check the purity of the drug, whether it is really pure active component or if there are any other contaminants in it. And also you can check the pharmaceutical samples. You can check the pharmaceutical samples, and also you can check the food samples for adulteration. You can check the food samples for the adulteration. What is adulteration? If you have any unwanted contaminants in the food, which will degrade the quality of food, then they are nothing but adulterants. You can even trace the adulteration of food samples. Now we will see what is this monitoring the progress of reaction. How to monitor the progress of a reaction. How can we do that monitoring of the progress of a reaction? Suppose I am taking two reactants A and B. I have taken two reactants A and B. They are combining to give you a product C. A and B are the two reactants which are combining to give you a product C. Suppose this is our reaction class, where we are I have added all the reactants and I have kept the reaction conditions. Everything is set up. So initially I have taken a sample with the help of a capillary tube. Then I have placed it on the origin. Then I have done the development by keeping it in a solvent system. So after separation I will get two spots. What does that indicate? That means both the reactants, they are still there. So after some 20 minutes I will take the reaction mixture. After 20 minutes of adding the reactants, first I have taken immediately, then after 20 minutes I am taking the sample with the help of a capillary tube. I am placing it on the origin. Then I have done my development over here. So if I see three spots, what does that indicate? Product is in the state of formation. The reaction has initiated and the product formation has also started. Now after some 60 minutes, I have given it ample of time. Then I have done my paper chromatography on it. I have taken the sample from here after 60 minutes. 1 hour I have taken the sample. I have put my sample over here with the help of capillary tube. Then I have introduced it into the mobile phase. Then if I get only one spot, what does that indicate? Product is formed. The reaction is complete and the product is formed. Henceforth, you can see only one spot which corresponds to the product C. So, in this way, paper chromatography, even it is a simple basic technique, you can do so many applications on it. You can separate the components, you can check the purity of the sample, which is very, very important nowadays. Then you can also monitor the progress of a reaction, or you can check the completion of a reaction. Is it clear, girls? Yes.